Thank you so much. Good morning, everyone. My name is Mathura, and I'm a master's student in the Department of Chemical Engineering here at McMaster University. I would like to thank DDL for giving me the opportunity to present our work investigating the effects of fluid composition on bacterial aerosol production. More specifically, this talk will address the relevance of airway mucus composition for infectious aerosol production. So the ongoing COVID-19 pandemic has generated a lot of interest in airborne transmission of respiratory infections. Airborne transmission requires three things: the generation, inhalation, and deposition of infectious particles within the respiratory tract. These particles are expelled into the environment during normal respiration or as an individual speaks, sneezes, or coughs. Biological particles are polydispersed in nature. Importantly, finer aerosols less than 5 micrometers in aerodynamic diameter can stay for hours due to slower settling velocities, whereas larger aerosols greater than 5 micrometers in diameter will sediment very quickly due to faster settling velocities. This makes finer aerosols much more likely to be inhaled as compared to larger aerosols. Infectious aerosols can be produced through the vibration of vocal cords, the breakup of the airway mucus lining, and due to the reopening of collapsed terminal bronchioles at the start of inspiration. Today, we will focus on the second mechanism, which is depicted on the image here. Specifically, high-velocity airflows generated within the airway lumen during respiratory activities can lead to shear stress-induced breakup of the mucus lining along the air-mucus interface. Pathogen-containing particles can be depicted within the red circle here on the image. Given that the airway mucus can be a source of infectious aerosols, it is therefore important to understand its composition. The airway mucus lining is a complex hydrogel composed of 98% water and 2% solids content. As seen on the image to the left, the mucus layer sits on top of the periciliary layer, which coats the ciliated epithelial cells of the airways. Solids concentration is described as the dry weight percentage of solids per given volume, and as shown on the image on the left, is primarily made up of salts, proteins, and mucin glycoproteins. Expelled mucus collected from healthy individuals, as well as those with airway disorders, were analyzed for solids concentration. Importantly, solids concentration was shown to increase with disease severity. For example, increasing from 1.7% solids to 3.7% solids in chronic obstructive pulmonary disease and 7% solids in cystic fibrosis. Given these conditions, we wondered whether altered mucus concentrations can influence infectious aerosol production. To investigate this, we related physiological parameters of bioaerosol production to our experimental condition. Peptone water is a common microbial growth medium rich in salt and protein content, similar to the airway mucus. We adjusted the concentration of the peptone solution to reflect airway mucus concentrations in health and disease. Next, we suspended the bacterial pathogen Staphylococcus aureus, which is known to colonize the airways, in Peptone water, achieving a final concentration of 5 × 10⁴ colony-forming units, or CFU, per ml. To generate infectious aerosols, bacterial suspension in Peptone water, as well as compressed air, was delivered to the Blaustein atomizer. We specifically selected the Blaustein atomizer as it can be operated in single-pass mode, which does not concentrate the feed solution. Polydispersed bacterial aerosols are generated as high-velocity compressed air exiting from the nozzle of the atomizer breaks up the liquid. So, to provide a bit more detail, the full experimental setup can be seen here, which has been designed in accordance with the ASTM F2-101-19 standard specifications. Specifically, the bacterial suspension is delivered to the atomizer at a feed flow rate of 200 microliters per minute. At the same time, high-velocity airflow is supplied to the atomizer at an airflow rate of 1.5 liters per minute. Aerosols are generated by the atomizer positioned at the top of the 60-centimeter chamber. These aerosols become diluted by air drawn in through the HEPA filter found to the right of the chamber at an airflow rate of 26.8 liters per minute. At the base, the aerosol mixing chamber connects to the inlet of the impactor. A downstream vacuum pump is then used to draw aerosol-containing air from within the chamber down into the 6-stage cascade impactor at an airflow rate of 28.3 liters per minute. During a single experimental run, the atomizer is operated for 1 minute while the impactor is operated for 2 minutes. Returning to our objective, we set out to examine the effect of peptone water concentration on bacterial aerosol properties by examining the aerodynamic size distribution, mean particle size, and total particle counts generated. As pictured on the left, a 6-stage cascade impactor loaded with tryptic soy agar plates was used to collect bacterial aerosols. Tryptic soy agar is a common nutritional medium for culturing bacteria, including Staphylococcus aureus. After aerosol collection, agar plates were incubated at 37 degrees for 20 to 24 hours. I want to draw your attention specifically to the yellow bacterial colonies forming concentric circles on the image on the right. These colonies are produced by bacterial aerosols, which have traveled through stage perforations to impact on the collection plate below. The aerodynamic size of bacterial aerosols was determined using the stage cutoff diameter, labeled in white, while bacterial aerosols were quantified using colony counts. Moving on to the results, first we examined the aerodynamic size distribution of bacterial particles collected across the 6 stages of the impactor. The fraction of total particles collected on each stage is shown on the y-axis, while the aerodynamic diameter is indicated on the x-axis. Greater fractions of bacterial particles generated from a 1.5% peptone solution were collected on lower stages with a cutoff diameter less than 3.3 micrometers. On the other hand, greater fractions of bacterial particles generated from a 10% peptone solution were collected on upper stages diameter above 3.3 micrometers. Next, we examined the mean particle size of bacterial aerosols. On the graph, mean particle size is depicted on the y-axis with peptone water concentrations represented on the x-axis. As indicated by the red arrow, a 1.5% peptone solution produced bacterial aerosols with significantly smaller mean particle sizes. On the other hand, a 10% peptone solution indicated by the blue arrow produced bacterial aerosols with significantly larger mean particle sizes. Lastly, we examined the total bacterial particle counts generated. Once again, on the graph, total particle counts are depicted on the y-axis with peptone water concentrations represented on the x-axis. As indicated by the red arrow, a 1.5% peptone solution produced significantly greater number of bacterial particles, whereas a more concentrated peptone solution, indicated by the blue arrow, produced significantly fewer bacterial particles. So, to summarize our results, we have experimentally demonstrated that changes in peptone water concentrations affects aerodynamic size distribution, mean particle size, and total particle counts of bacterial aerosols. Importantly, we have conducted these experiments using a bacterial species known to colonize the human airways, suspended in solutions formulated to reflect physiologically relevant airway mucus concentrations. With regards to clinical applications of our finding, it stands to reason that changes in the composition of the airway mucus lining may lead to altered infectious aerosol production. Further exploration of the link between airway mucus composition and infectious aerosol generation is needed to better understand airborne transmission dynamics. So while bacterial pathogens such as Staphylococcus aureus are approximately 1 micrometers in diameter, viruses are an order of magnitude smaller with diameters of 0.1 micrometers, as is the case with COVID-19. Therefore, our next steps will focus on the study of viral aerosol properties using bacteriophages or bacterial viruses as surrogates for mammalian viruses. Understanding the influence of biological variables on bacterial and viral aerosol properties will allow us to conduct filtration testing of personal protective equipment, such as masks and respirators, under more physiologically relevant conditions. Thank you so much for your time and attention today. I welcome any questions at this time.
Maddie, that was super. Thank you so much. Very clear, very nice slides. Thank you for taking us and sharing your journey with us. We do have quite a few questions that have come through, but if I just start off, maybe Michelle, And if I ask you, Maddie, just a little bit about your methodology. So the Blaustein atomizer, why did you choose it? And what were the challenges when you were setting up the methodology? And what are the you've shown us some of the advantages of the atomizer, but what were the disadvantages? So a little bit about the challenges that you faced when trying to get your model to be a good model.
So the main reason why we chose the Blaustein atomizer is because it is single pass. So once a solution reaches the nebulizer, it aerosolizes that liquid as opposed to recirculating it. So when it comes to the Collison nebulizer, which is the common alternative, it constantly recirculates the solution. So the solution, there's a little bit of evaporative effect, so it concentrates it, which we don't want. In terms of challenges, when you operate an atomizer, you have to choose the airflow rate very carefully, because when you increase the airflow rate relative to the feed flow rate, you generate smaller aerosols. And when you decrease the airflow rate relative to the feed flow rate, you increase the size. So because we are following the ASTM standard, we have to narrow down the parameters to achieve the ideal size of aerosols.
No, Maddie, thanks very much. No, and that makes a lot of sense there. I've got lots of other questions I could ask, but Michelle, I'm going to hand over to you.
Yes, thank you very much, Maddie. Now, we've had a couple of questions come in, which are really relating to the technical and practical aspects of aerosolization. So maybe I can give them a couple of parts. One question was very much around your cascade impact setup. Is a cascade impact with agar plates sort of standard in-house modification? And how did you recalibrate the effective cutoff diameters, for example, within the cascade impaction with these modifications in place?
So the cascade impactor that we're using is a viable six-stage cascade impactor. So we didn't actually have to modify the cutoff diameters. We're using what is specified in the manual to determine the mean particle size of the aerosols which are collected beneath each stage, using the triptych soy agar plates.
Okay, thank you. And the second part of this question was around the aerosol mixing. How did you ensure that there was uniform mixing, and so actually the deposition pattern was homogeneous across the aerosolized particles?
So the way we achieve this, once again, because we're following the ASTM standard, we're relying on that 60 centimeter chamber to allow the diluting air to come in and mix before it reaches the cascade impactor at the very base. So as the aerosols travel from the top to the bottom of that chamber, it has been well mixed before it reaches the impactor.
Okay, thank you. Omar, do you have any more questions?
Yeah, so Maddie, we've got lots of questions, but obviously we've only got a limited amount of time. So what advice would, so the future application, so you're probably going to come to the end very soon and somebody will take over your work. How would you see this work progressing further?
So the immediate goal is to make it more relevant to the current ongoing pandemic. And the main thing is the size of the pathogen that we're encapsulating within the aerosol. So currently, all these tests were performed, and most commonly they're being performed with bacteria, which are 1 micrometer or an order of magnitude larger. So we need to use pathogens which are of a similar size, shape, and charge as COVID-19, which we can actually do quite well with bacteriophage 5-6, which is the next step to this project.
Okay, thank you. Now we've got just 1 minute before the end of this session, and I've had a question come in that I think is really interesting. I'd like to ask if we have time, and I think it relates to what you were just saying earlier, Maddie, and it was around, what I think I was seeing is that you are seeing an impact of the peptone loading on the particle size of the droplet. And so the question is, well, how do these findings actually then translate into the relative risk of disease transmission across a patient population with different degrees of muco-obstructive airway disease?
It's hard for me to comment on that. It's very much explorative. So I haven't tested any clinically relevant samples. in general, what we find in cases like cystic fibrosis, there's a thickening of mucus. So because of that, the expectation is that denser particles will be generated, and they will settle more quickly to the ground as opposed to being in the air for much longer, increasing the risk of being inspired by a person in the vicinity. However, it is very difficult for me to comment on the clinical relevance without conducting or repeating these experiments with clinical samples, mucus samples from patients.
Absolutely. Thank you very much, Maddie. That was a really interesting presentation. So very well done on your work.